right, welcome everyone. I'm sorry if you had difficulty making your way into Mission Bay. I understand there's a disaster at the intersection here, but thank you for being here. So I think I know most of you, but for those whom I don't know, I'm Sandy Williams, Gladstone President, and I'm very happy to have you here for our fall symposium. So as some of you know who are veterans of this event, we do this twice a year. It's a chance for us to bring our friends and supporters together to share what's new and interesting and exciting about Gladstone, and also to get to know you a bit. So please, when we go to the reception period, find Gladstone people wearing their name badges and let us get to know you even better. This is a special symposium for me. I've enjoyed presiding over these for a number of years now because, in a way, this is my valedictory. As many of you know, if not all, this will be my last symposium as president of Gladstone. I have the great joy and privilege to have been able to choose my successor, find that successor right here with me, and then spend several months preparing him to take the job. And where is he? There he is. Deepak Srivastava. Yeah, I saw him a minute ago, and then I didn't see him. But it really is, how fortunate am I to have had the privilege of working at Gladstone for all these years, but then to get to manage this transition in just the way that I wanted to, and it's going very well. So effective January 1st, Gladstone will have a new president, and that's a bit sad for me to say that today, but not so sad. One, the time is right, I'm ready, and two, I'm not saying goodbye. So the plan is for me to stay on in a part-time role for Gladstone, serving him, and the good news is that'll give me opportunity to still be together with many of you over time. But with that, I'd like to invite our president-elect, and this is his first public appearance as such, to come to the stage and say a few things.
Well, thank you, Sandy, and thank you all for being here today. I want to start off by thanking Sandy for carrying us through these last 8 years. With the economic times when just before he came in, was a rocky period, and Gladstone has continued to grow and thrive thanks to Sandy's efforts. And I want to also acknowledge Jennifer, who moved out here to be with Sandy and has been a supporter and helped us get through these last few years and continue to accelerate the impact of Gladstone. And so, many of you in the audience I know very well, and I thank you all for your longstanding support and interest in what we're doing, and many messages over the last several weeks of congratulatories. But what I want you to know is that there are not many, maybe there's no other place that I would have taken this sort of job. This is a place that I've come to love dearly because for what it stands for. I don't think there's a single other institution in the world that brings to bear the level of science that we do here that is also so laserly focused on disease. And in each area where we have tried to impact human disease, we have made a tremendous impact. And that's because of the business model how we go about it, the culture of how we do things here, and that was really all set by Bob Maley, who's here in the audience as the founder of Gladstone and founding president of Gladstone. So please join me for a moment in thanking both Bob and Sandy for what they've built. And my hope is to be able to continue to make this a place where we are solving the foundational basis for some of mankind's worst diseases. We're not stopping there. We're going to, and it's a place where we take those findings and try to ensure that they actually get to people and try to make a difference. And finally, that we're a place that trains the next generation of leaders, because we'll all be gone at some point, and our greatest legacy will be those people that we train who leave here and go on to keep making great discoveries and impacting human life. So I'm honored to be the next president at Gladstone, and I look forward to working with all of you to tackling mankind's worst diseases. Thank you very much.
Okay, so now on to the main event for this afternoon, which is to get up close and personal with three of not only Gladstone's best scientists, but I would say the nation's best scientists. They're each going to give you a little window into the work that they've done. And there's a unifying theme that I'd like you to follow as you hear what they have to say and then have a chance to ask them questions. And the unifying theme is that science is unpredictable, it's full of surprises, and the great discoveries and breakthroughs will come to those who have the imagination and the courage to see what others haven't seen, to see a path to a cure through an unconventional route that was either unseen or rejected by others. And common to each of the presentations that you'll hear will be reports of leaps of genius and imagination that allowed these scientists to see a very uncommon and surprising path. So science overcoming disease begins with science, and in science you need to expect the unexpected, expect surprises, but then when you see those surprises and the unexpected appear, know what to do with it so you make progress where you want to go. Now, I'd ask you to look in your program. I'm not going to introduce the scientists from the stage up here. I'd ask you to read about them in the little summaries that are here. And the format here will be, I'll ask each of them to come up and tell a story about their science. They'll take less than 10 minutes to do that each. And we won't ask them questions until the very end. But after the three of them have told the stories to you, I'll have them come up here and we'll open it for questions and answers, and I'll moderate. So with that, let's get going. Our first speaker is Melanie Ott.
Well, thank you, Sandy. And thank you to all of you for coming out and supporting our science here. So we are starting with the flu season. It's October, November. So who of you had their flu shots already? Well, very good. I'm really impressed. I hope the rest of you will take example and get it soon. Although we all know that some of us will eventually come down with flu-like symptoms. And we will, especially at the beginning, not know whether it's really the flu or if it's another cold that we have caught, caused by other viruses. But it would be good to know if it's the flu, because we know that the flu is serious and we should see a doctor. It would also be good because there's actually a drug against the flu that we can take very early. And when we take it very early, we can actually help making the disease less severe. Wouldn't it be great to not worry about whether it's the flu or whether it's another virus that we have, and just take a pill against our flu-like symptoms and hit all of them, the viruses that cause the common cold, the influenza viruses that cause the flu, and not to worry exactly what virus we're dealing with. And the same thing is with bacteria and viruses. We all know about sore throats. They also will come pretty soon now in the next few months. And we will not know whether we will have a strep throat caused by a bacteria or whether we have a viral infection. But we very well know that if we have a strep throat, we should take antibiotics. And we also know if we have a viral sore throat, we should not take antibiotics, because that will actually make resistance development among the bacteria much more severe. So again, wouldn't it be great if we don't have to worry about, is it a streptococcus or if it's another virus that is actually causing our symptoms, but just be able to take one pill and help with our symptoms and make it better. Now, we here at Gladstone think it would be great, not only because it's convenient for you and us to have only one pill and not to worry about it, but we also think that it makes scientific sense. And I will come back to this actually at the end of my very short presentation. So the approach that we are taking here at Gladstone is that we are not going after each individual virus and bacteria to try to develop a drug against it. What we're doing is we focus instead on the response in our body that actually these pathogens evoke when they come and infect us. And it's actually not so easy for a pathogen to set up shop in our body and to cause an infection and make us really go sick. There's millions of alarm bells ringing when these pathogens are coming close to us and starting to invade us. And this is not only all the immune cells that are, directed directly to attack any infected cell or any bacteria that is around. In each of our cells that compose our body, there is actually there are programs hardwired in that are just dedicated to sensing, pathogens that are coming in and then triggering defense mechanisms. And these are usually suicide mechanisms that lead to the killing of that cell. Now, the pathogens are also not, they're not bad. They actually have evolved to circumvent all these alarm mechanisms with more or less success. And they either sort of try to circumvent these defense mechanisms, or they try to inactivate them, or they tried actually to hijack them so that they help their own replication and not really do their job in defending. So it is a very complicated and complex interaction between the pathogens and our body that leads eventually to a infection. And what we're doing here at Gladstone is that we're trying to unravel comprehensively this complicated network of interactions that are occurring. So what we're doing is we're expressing thousands of proteins, viral and bacterial proteins. We study with what cellular factors or host proteins these viral proteins come into contact. So usually one protein interacts with tens or hundreds of our own proteins. So we're dealing with hundreds of thousands of interactions from which we extract those common pathways that are actually common to multiple infections, bacterial or viral. And then we study whether these pathways are important for the infection, so we should actually inactivate them with a drug or a cure, or whether these are actually defense mechanisms that we could strengthen so that they could work better against multiple bacteria or viruses. And using this approach, we have confirmed already, also we're still in the process of doing this, that pathways that should be involved are actually coming out in our analysis. And we can now determine which of the viruses and bacteria we're studying are actually depending on these pathways. But we're also discovering a lot of new and unexpected pathways in our body that before have not been brought into contact with pathogens. For example, we recently identified a genomic surveillance pathway that was previously only known to target our own genome integrity, to make sure that our genetic information is correctly translated into proteins and is not mutated and can cause cancer or something else. So we have found out that surveillance pathway is also very effective against genomes that are incoming from pathogen and can recognize those and lead to very effective defense mechanism. And we have identified groups of viruses that actually are targeted by these genomic surveillance pathway. And among these is the Zika virus that I'm sure you all have heard about. Now, Zika is interesting because Zika has evolved a mechanism to inactivate this pathway in order to set up shop successfully in our body. By inactivating this pathway, it not only allows itself to replicate, it also harms our body, because that genomic pathway is very important for us. And it's not only important for cancer development, we find that it is also important for microcephaly development in developing brains. So I think our approach currently is very ambitious and it's very risky. And drug companies normally shy away from such an approach because it's a little bit like finding the needle in the haystack. An argument is also that by targeting not the pathogen, but actually targeting our own body, we risk to have more side effects by generating a drug, and that might be right. However, we think that the advantage that comes with this approach is equally important, because by targeting not the pathogen, but targeting our own body, the pathogen cannot develop any resistance. So we're dealing, or we're having actually an angle on the growing resistance problem that we have currently with antibiotics. Resistance is not only an issue with antibiotics, it's also important with antivirals. I'm sure you all have heard about the hepatitis C drugs that are so effective but also very expensive. One reason why they are very expensive is because they're not only composed of one drug, they're composed of 2 or 3 drugs at the same time, because if we only use one drug, the virus would develop resistance immediately. Another advantage that we see in this approach is that we don't really need, if we're dealing with an emerging epidemic, an unknown virus that is going to come or here or somewhere else, we would not have to wait until we have all the last characteristics of this pathogen. We would have an arsenal of drugs that would be effective against multiple of these pathogens, and we would have a much better handle on these emerging infections. And as a last point, I want to point out that it is just an enormous advantage in basic research advance that we will gain out of this approach. Not only will we understand better how pathogens are going to interact with our body, we think that we will actually get a lot of information out of it that is also relevant for other diseases, like cancer or neurological or cardiac diseases that we will hear about in a minute. Because many of those involve chronic inflammatory conditions, which is basically a condition that starts potentially with an interaction between a pathogen and our body, and then the defense mechanism can get out of control. So by understanding better how these mechanisms work, I think we will also contribute to other diseases. Thank you.
So just as a postscript to what Melanie said, I think many of you are aware that in the world of cancer medicine, very hot and current, that drugs have been developed that don't go to try to kill the cancer directly, but help our own body's immune system defeat the cancer. That's not exactly the same as what Melanie's described, but those are targeting our own proteins to resolve the disease. And here, Melanie and this very formidable team she's put together is taking a similar approach to protect us from MRSA and drug-resistant tuberculosis and influenza and the other things that threaten our future health. Very exciting. Our next speaker is going to be Saptarsi Haldar, or Sap as we know him. And Sap, the floor is yours.
Thank you, Sandy, and I'd like to thank all of you for being here and for being friends of the Gladstone Institute. So for those of you that don't know me, I'm relatively new to San Francisco and to the Gladstone. I moved here 2 years ago to join the Gladstone Institute as a scientist on faculty here studying heart disease, and I'm also a practicing cardiologist that takes care of adults with heart conditions here in San Francisco. And so I'm going to tell you today about some work that we're doing for a condition called heart failure that many of you may know. So who here has heard of heart failure or knows somebody with heart failure? So not surprising, I see a lot of hands here. So before we talk about the disease, I think I'm going to tell you a little bit about how amazing all of our hearts are. And I know it sounds kind of obvious, but I think it's important to just reflect on this. So the job of your heart is to pump blood to every single cell and organ in your body. So if the heart can't do its job, every single part of your body suffers and deteriorates. So it's absolutely central. And so the heart is about the size of your fist, as you all know, and it's made up of billions of little heart muscle cells that are collectively all work together to pump blood. And when the heart doesn't pump blood well, bad things happen. As I told you, all your organs don't work. The liver doesn't work. The kidneys doesn't work. The brain doesn't work that well. And it's a miserable existence. People feel lousy. They can't do things that they should be able to do, like walk up a flight of stairs, walk in the grocery store, do some little bit of exercise. They have trouble sleeping at night. They fill up with fluid in their lungs and their legs, and people feel like they're drowning. And it doesn't get better. It just progresses and progresses. And to make an analogy here, imagine, it's a little bit of a loose analogy, but I think it's appropriate. Imagine that your heart is like an engine to a car, and we're all born with, say, a 8-cylinder engine, if you have a normal heart. And the heart does its job, but over time, things can injure the heart. So if you have a heart attack and part of those heart muscle cells die, or you have problems with your heart valves and the heart gets overloaded, or you have high blood pressure for too long and the heart has to work too hard for years and years and years, what happens is that slowly you go from an 8-cylinder motor to a 6-cylinder motor. And some people have a 2-cylinder or 1-cylinder motor if they lose enough of their heart muscle cells. And that's a really bad thing, because a car, if it's malfunctioning, you can keep it in the garage, but your heart has to keep going, right? It has no choice. And so imagine your heart has to keep beating every single day of your life on 2 cylinders. So it's this vicious cycle, and the heart just keeps getting worse and worse and worse. So we've done really good with a lot of things in cardiovascular disease over the last 40 years. As a field, we've done really well with heart attacks. We've done well with, we can open up heart arteries with stents and do bypass surgery. We've done well with lowering people's blood pressure and cholesterol, thanks to the work of Dr. Maley here and many others. But we haven't done so well with fixing the actual motor when it starts malfunctioning, this vicious cycle of heart failure. And so that's what I'm going to talk to you today about new ways, and maybe a surprising way that we found that is able to treat heart failure. So what's the current state of affairs? So heart failure is common and it's lethal. So do any of you guys have any idea how many people in the United States have heart failure? Any guesses? It's very high, but it's 6 million people in the United States, and it's growing. So to put that number in perspective, that is larger than most major metropolitan regions in the United States. The Bay Area is estimated to have 7 to 8 million people in total. So this is a large number, and it's lethal. So if you get diagnosed with heart failure today, in the next 5 years, your chance of being alive at year 5 is a coin flip, 50%. And so despite current medical therapy in 2017, those are the numbers. And that's worse than some of our most common cancers: breast cancer, colon cancer, prostate cancer. So we really have to do better for our patients and our loved ones. And so what do we use in this day and age? So many of you may be familiar with these drugs or heard some of these drugs. We use drugs that basically block fight or flight stress hormones like adrenaline to kind of keep the heart from being bombarded with these adrenaline-like hormones, because that's what happens when the heart starts working on 2 cylinders. The body overreacts and pours out adrenaline, and that creates this vicious cycle of damaging the heart. So we use things like beta blockers, some of you may have heard of, or ACE inhibitors like lisinopril, and those have done okay, but remember, people are still dying 50% at 5 years, so we have to do better. So all of these drugs work by blocking these adrenaline hormones at the surface of heart muscle cells. So we reasoned that could we actually work with drugs that go deep inside the cell and get into the nucleus of the heart muscle cell, the so-called command center of the cell, to somehow change the software or the programming of the cell to kind of make the heart muscle cell better, right? And so we took a surprising approach here, and this is kind of when you keep your antennas up to different areas of science and you hang out with other scientists, you start to pollinate your own ideas, and things kind of catalyze. So when I was training in cardiology in Boston, I was good friends with a scientist named Jay Bradner that many of you guys know, and he is an oncologist and was very interested in finding ways to treat leukemia. And what happened is he was telling me, and eventually published, a paper that showed a prototype drug. It's called JQ1. You don't need to know the name, but it's actually named after one of his students, Jun Chi, that synthesized the compound. And the way this compound works is, to make a long story short, it actually goes into the command center or the nucleus of a leukemia cell, right, and tricks it. So Jay asked the question, well, how does a leukemia cell, that cancer cell, remember how to be cancer, every time it divides? And it turns out that there's machinery within the nucleus of the cell that tells the cancer cell how to be a cancer cell, to keep growing and dividing and taking over the body. And he created this compound to literally make a leukemia cell, this cancer cell, forget how to be cancer. And this compound is now in a number of clinical trials for a number of cancers, including leukemia. So we'd heard about this, and I asked myself the question, kind of a light bulb went off, is that, well, if you can make a cancer cell forget how to be cancer, can you make a stressed out heart cell that's malfunctioning, can you kind of make it forget how to be sick in a way? Right, and so that was the initial seed idea. So I remember that I called up Jay Bradner, and I asked him, you got to send over some of this compound, we have to try it. And I still remember when we did this experiment. So I had a medical student in my laboratory as a summer medical student. I was like, you should try this. So what we did is we took this compound JQ1 and we put it on heart muscle cells grown in a dish that we made to be stressed out like they were in heart failure and sick. And we looked at the cells after we'd given the JQ1, and we saw that actually the heart cells looked much better after as early as 24 hours. And so that's great. That's heart failure in a dish. And so it's great. But we want to know, does it really affect heart failure in a living creature with heart failure? And so the next step, as you know, is that we use animal models. So there's animals, and we use a lot of mouse models that have heart failure. And so we did a clinical trial in mice with heart failure, and we gave one group a placebo and one group the JQ1 over the course of 1 or 2 months. And we didn't know which group got which. It was a random study, and the trainees in my lab didn't know which animals got which when we were looking at them. And just like we do for patients, we can do ultrasounds in these little mice and look at the hearts and see how they're pumping. And I still remember the day. We didn't know which group was which, but my graduate student told me, Sap, I think we've been doing this for about a month. He's like, there's one group of these mice, when we're doing the ultrasounds, that their hearts actually are pumping really good, and the other group is getting really bad. And so we got really, really excited, and when we finished the experiment and we kind of unblinded the result, we'd realized that the JQ1 compound had been really healing or preventing these hearts from failing. It was really striking. It's one of those exciting moments in science when you know you're onto something kind of big. And so we've been really interested in knowing how this works in heart muscle cells. And we're still trying to figure this out, but I think the way we're thinking is this. So if you think about how cancer works, to make an analogy, cancer is kind of like the cells in cancer are like a rogue street gang. They're, they do their own thing, and anytime conditions get a little rough or times get rough, you give them a drug or you try to kill the cancer cells. You could kill one gang leader, but there's always another gang leader that comes up no matter how hard you try. And that's the real challenge with cancer. There's always that one gang leader that will try to come up and get resistant to what your therapies are. But the heart is different. The heart muscle cells are kind of good worker bees. In the sense that they don't try to take over the body and grow. There's no gang leader per se, but they all try to pump together. But the difference is that your heart is not an organ when you're an adult that grows, right? Necessarily tremendously. So what the heart does is it does the only thing it knows how to do. So our hearts is really evolved to do one thing really well, is to heal itself and with wounds when it's injured. So we're not meant to have big heart attacks or high blood pressure for 20 or 30 years, right? But what the heart is good at doing is if you got stabbed by a saber-toothed tiger 50,000 years ago, trying to heal the wound with scar tissue so you don't bleed to death, right? And that's kind of what the heart does when you injure it with high blood pressure or a heart attack. It heals the wound, and it tries to keep functioning. But the problem is these kind of wound healing responses that the heart have, if they're left to go on for too long, a period of time, or too high, if they're too revved up, they ultimately fuel the fire of damaging the organ. And that's really how these drugs like JQ1 work, is they turn down the dial these wound healing type responses, that really helps the heart heal over time. So where are we going this? We're trying to understand how these drugs work in the heart, and we're looking for partnerships with pharma companies and other institutions to try to partner with us to try to develop this and eventually try to go first in human for patients with heart failure. And the other thing I'd like to highlight for you is that, you know, if any of you guys have been listening to what's going on in cancer drug discovery, one of the big problems with cancer therapies, all the new cancer therapies that come out, is that a lot of them are good at killing the cancer, but a lot of them are quite toxic to the heart. It's become a big problem. So we're actually in a privileged space. We actually are dealing with prototype drugs, and actually drugs that are in clinical trials for cancer that are both anti-cancer, but actually can protect the heart, which is a really nice space to be in. And I'll leave you with the notion that this is not just a JQ1 thing, as we've discovered in the lab. So we're looking for other players that might be drug targets in the heart. And I feel that we're at the tip of the iceberg in deciphering this. And for every JQ1, there may be a whole host of other drug targets that live in the nucleus or command center of the cell that might work this way. So this may open up a whole different approach for heart failure. So thank you for listening.
So I've followed SAP's work very closely and admired it before we attracted him to come to Gladstone, and even more so after he's been here. But I have to admit, I had a moment of anxiety during his story when he said that he got this new drug into his lab and he wanted to test it, and there was a medical student. I thought he gave it to the medical student. I was so relieved that it was only a mouse, or it was only cells in a dish. Just kidding. We like to have some fun around here sometimes. All right, our next speaker is Katerina Akassoglou.
Thank you so much, Sandy, and thank you all for coming here. So when we think of the brain, we usually think of neurons, and these are the cells in our brain that help us think, help us sense our environment, help us communicate. These are the cells we use to remember. But our brain doesn't only have neurons. It also has a very intricate network of blood vessels. What you can see in the slide behind me is a human brain, and these are the blood vessels that we have in our brain. So what do the blood vessels in our brain do? And they do perform two main functions. One is to give all the food to our neurons. Our brain cannot survive without nutrients going inside our brain, and these blood vessels achieve this food reaching our brain. But they also perform a second very, very important function, and the second function is to form a barrier. And this barrier doesn't allow any toxic products from our blood to enter inside our brain. And this is how our brain can remain in an environment that allows it to do all its normal functions. When I was a graduate student and I was doing my thesis work, I made a very unexpected discovery. I was working with an immune factor, and what I found out was that this immune factor was opening holes in the blood vessels of the brain. And by opening those holes, it was basically breaking that barrier. So now blood proteins were not anymore restricted in these blood vessels, but instead were leaking all over the brain. And this leakage of blood proteins inside the brain happened to coincide with some of the worst death of neurons that was happening around those blood vessels. And I found it really fascinating that when blood was leaking inside the brain, that coincided with this very bad damage to neuronal cells. Now, of course, when in biology we have these coincidences, we always have the chicken and the egg question. Is it because we have more damage that we have more blood? Or is it possible that the blood is actually contributing to the disease. So the way that, the reason that I thought that this question was important to study and was important to dedicate my entire scientific career on was because of the impact it has in neurological diseases. The presence of blood in the brain and its correlation with neuronal damage and neuronal death is a common thread among every neurological disease. Multiple sclerosis, Alzheimer's disease, trauma, traumatic brain injury, spinal cord injury, different forms of epilepsy, all have a common feature, which is blood leaking in the brain. And what I study in my lab here at Gladstone is what does the blood do when it leaks in the brain. And what we have found in our research is three main functions of what the blood does when it finds its access in the brain. And this happens only in disease. And these three main functions of the blood, one is that it's inducing inflammation. Our brain, in its normal situation, has never seen blood before. It's always trapped inside these blood vessels. When it gets out of those blood vessels, it hijacks normal mechanisms in our brain and turns them to be pathogenic, what we call inflammatory. The second function that we found that the blood does is that it kills our neurons. So what it does, it creates an environment that is so toxic that our neurons cannot tolerate it. So when there is blood leakage, neurons die. And the third thing we found, which is hot off the press, it's a brand new discovery that we found that when blood leaks in the brain, it also stops normal repair mechanisms. It stops the stem cells in our brain, the progenitor cells that are there to repair after a damage, from actually being able to perform these repair processes. And instead, it's even worse, it turns them into cells that contribute to the pathology. It switches what we call their fate. So this really changed the way we think about blood in the brain, not as a secondary factor that is there as an epiphenomenon of the disease, but actually being in the driver's seat, driving the pathology. Now, all this might sound really dramatic, of blood leaking in the brain and causing all this pathology. And images like this, we thought, doesn't do it justice. So what we wanted to do was, can we use advanced technology and advanced imaging technology to actually see what I just talked to you about in the brain in real time? Can we actually see a blood vessel in the brain leaking in the real time and see how the cells respond to it? So here at Gladstone, we managed to develop advanced microscopy and used really powerful lasers and really powerful microscopes so we can see exactly what happens to a single blood vessel in our brain if something injures it. And that's something we have used here is the laser. You can see it in yellow. You can see the blood vessels here in red. And what you can see in green is these immune cells of the brain I was talking to you about. So if we play this movie, what you can see is this laser poking this blood vessel. You can see in red the blood leaking out of the blood vessel, and the cells in the brain in real time, within minutes, responding by activation to this injury. So now we have the tools to be able to see this not only in this acute model, but actually over time in different diseases. And we can study not just snapshots of how disease develops, but see in real time the whole sequence of events of the development of neurological disease, and at what stage leakage of blood is really important to contribute to disease. It is not a medical student. So, but thank you for the question. So how can we use this information that we have got from basic research and also from our imaging technologies for therapies? Is there a way that we can block these processes of blood in the brain? And if yes, will this help neurological diseases? Now, this was a very challenging question to answer because these proteins that when they get in the brain are toxic, unfortunately or fortunately, they actually have many beneficial functions when they're in the periphery. They become toxic when they get in the brain. So we had a big challenge. Can we selectively target them in the brain, but without affecting all the beneficial functions they do in the periphery? And what we achieved is to develop the first pharmacological tools to actually selectively stop these proteins in the brain without affecting any of the beneficial functions peripherally. Our first tool is a monoclonal antibody that can actually go and do exactly that. Stop these proteins from what they do in the brain, but without affecting their protective functions, in this particular case, in blood clotting, which is really important that we maintain during disease. We have very promising studies in our animal models, and these are animal models of multiple sclerosis, animal models of Alzheimer's disease. And we're very excited to continue this across the board of neurological diseases because these mechanisms are, as I mentioned, the common thread for many neurological diseases. So where we're now, we're at the cusp of really rethinking the way neurological disease develops. This is very timely. As you probably read in the news, many of the clinical trials are failing in neurodegenerative diseases. Antibodies against amyloid, there are several trials from many companies that don't seem to work in patients. It is timely and it is urgent for new hypotheses for the study and for the treatment of neurological diseases. And when we think out of the box, the way that the blood harms the brain is one of those new hypotheses that has the potential to change the way we think about neurological disease, but also change clinical practice. Thank you.
So I'd like to invite all three of our speakers to come up and take their seats, and we'll invite questions. So just to reiterate for a moment to focus you, we've got very promising new threads of science that promise to provide us with ways to treat multiple infections with a single drug that could be used in a way that doesn't provoke resistance from the infecting pathogens. We've heard of a completely unexpected and surprising route to correct the heart failure state by acting on the genome inside of the nucleus. And we've heard the surprising result that simple blood proteins involved in normal clotting, if they gain access to the wrong place in the brain, do much damage, and there's a way to apparently to stop that. So this is Gladstone at its best, science on a path to overcoming disease. So I got to applaud that again. But now here you are, here's your chance, front row seat, to ask other questions of these experts. Will?
The rise of CTE in football players and so on, is that a function of the blood in the brain?
We're getting the material actually now. We have contacted the scientists at Yale to get us the material. There's obviously blood-brain barrier leakage, and we would like to characterize it. A lot of the symptoms, the cognitive decline, definitely are in line with what we're observing.
Sharon?
Thanks. Several of us have a friend with cancer who underwent, all the chemo, radiation. It very nearly killed her and apparently had no effect on the tumor. And she has since then had 3 infusions of an immunotherapy drug to do the kind of thing Melanie's talking about. It has caused her tumor to melt away, the doctor's words. When is immunotherapy going to start being done first, before they practically kill you with radiation and chemo, instead of as a last ditch? effort?
I think the FDA approvals are on their way. Actually, it's a good timing. I think it's just a matter of time, and efficiency to really know which cancers are responding the best to these, immunological therapies, and there's still a lot of exploration done. However, I think the first, applications are being approved, so that is only the start. And I think at one point it will become the drug of choice first before the chemotherapies. But it has to go. Unfortunately, it's ordered process of really proving efficiency and little toxicity, and then it will become the drug of choice.
Yeah, I have a personal connection to the immuno-oncology story in that I played a small role in the development of the first successful immuno-oncology drug that Bristol Myers produced a few years ago, and that's opened a floodgate now of follow-on drugs. But the way cancer drugs are approved is you first give them to the most desperate patients, and then as they show success, the FDA allows you to give it earlier and earlier in the process. But as Melanie said, this class of drugs, and there are now many more coming on, have been so successful where they're going to move to frontline therapy.
And do you estimate that as being 1 year, 5 years, 20 years?
Oh, it's always hard to estimate those things, but this is perhaps the most rapidly moving field of medicine. So I think, and the urgency of many of these previously intractable tumors will drive it pretty fast. Another prediction I'd make is once you see these things starting to work, these completely novel approaches to heart disease, infectious diseases, and neurological disease, once the first one works, you're going to see the same kind of floodgate open, and that's what we're trying to start here at Gladstone.
It's worth noting that, as I think you're alluding to, that the same principles in the immunotherapy from cancer is what Melanie was talking about applying to infectious disease, can be applied to inflammation, can be applied to autoimmune diseases. So there's tremendous potential for modulating the immune system for a whole host of human diseases. So it's just the tip of the iceberg.
One more comment. It's also important to point out that the original science that led to the discovery of the immuno-oncology drugs was not actually conceived as cancer research. It was basic immunology of how T cells work. And there was, at that point, no real insight that it would become what it did. In fact, many were skeptical and said it wouldn't work. So there were some bold pioneers who pushed it against the grain, and then now we have a great success.
Well, it was actually done a lot in HIV research at the beginning, and that has been met with a lot of skepticism at the beginning, that we would engineer our T cells to become effective, to kill the infected T cells, and it has proven to be correct, and now it's being transferred to many other diseases, including cancer, very successfully.
Chuck, hi.
Hi. Thank you for this. This has been very informative, and I appreciate it. I'm not sure I want to live forever, but it's encouraging. The question I have is, it seems that the FDA has been very slow in our country in accepting some of the new versus Europe, et cetera. And I wonder, are we getting a little better at that venue and moving a little faster in our bureaucracy in Washington?
Anybody want to take that? And I've been reminded that it's a good idea for us to repeat the question before we answer it.
And so the question is a great one, is what can we do to accelerate the process of getting drugs into patients and new therapies into patients and navigating the regulatory agencies such as the Food and Drug Administration in the United States? And there's a sentiment that things may be faster outside of the United States, which is in a large part correct. So this is a really complex question, and I think we can have a discussion. I know Sandy knows a lot about this as well as my other panelists. But the issue becomes, I think, the United States regulatory agencies are very concerned about safety first and do no harm. And there has been some experience over the last 5 decades with drugs inadvertently causing birth defects, for example, in the 60s and 70s. And later on, drugs that were initially prescribed to treat heart disease, for example, were later found to have more harm as we learn more about them. So there's a culture of caution. And it is a double-edged sword because what ends up happening is that we often delay the process of things that work. So I think what we should be doing is taking this question and having a streamlined process for really getting drugs first in human. And I think the leaders in this field are in cancer. I think in the cancer field, the pipeline from going from experiments in the laboratory to getting a first-in-class drug to getting drugs to patients who are refractory to all other therapies in cancer is much quicker. And I think all of the other fields that we're talking about, whether it's inflammatory diseases, cardiovascular diseases, and neurodegenerative diseases, should learn from that playbook. And I think there's a movement to do this in all of our fields, is to go first in human quicker. So do you have any other comments?
So a reassessment that I see in the testing of drugs for approval is a discussion around the reliance we have in animal models. So a lot of the decisions we make on whether a drug will or will not move to the clinic is how it does in animal models. And some of these animal models reflect the human situation, some though reflect only aspects of it. And a lot of the failure we see in the clinic has to do that the animal models are not really fully representative of the human situation. And now this becomes really problematic when you test a new hypothesis. So if an animal model is developed with an old hypothesis, it responds to the drugs of the old hypothesis that have not worked to the clinic. And when you have a new hypothesis and you test it to the model of the old hypothesis, it doesn't work to in the model, but it might have a really good chance to work in humans. So here at Gladstone, we had the previous commissioner of the FDA who met with our faculty. And we had this exact discussion about the circular problem of success and failure, and how can we break this reliance to the animal models. And I see a lot of reassessment and discussions about how important it is to really look at the human disease and how we can build hypotheses of from the human disease and how experimental medicine can actually assist for these out-of-the-box ideas a lot more than outdated animal models.
And perhaps you might want to comment.
Just one little comment I can just say. In this respect, we can probably learn very much from the HIV field again, because there was a lot of advocacy that has, happened very persistently and has led to the, accelerated approval of antiretroviral drugs. At that time, really a lifesaver for many patients. And I think it has to be sort of a good mixture between, making sure that nothing gets prematurely approved so that no harm is being done, but also not to, unnecessarily delay any approvals because of bureaucratic hurdles. And I think advocacy is something that can help here.
And to get back to this point of models to study therapeutics, and it'd be a good juncture to remind everyone that the Gladstone has really pioneered learning about human disease using human stem cells and patient-specific stem cells that we can grow and start to understand how disease occurs in humans and test approaches. And I think all the investigators at the Gladstone leverage this technology. And so that's another complementary approach that a lot of us are trying to use to create better models to understand human disease and develop new therapeutic approaches.
Yeah, and maybe one more comment how this can come together. Obviously, animal models are very important for aspects of the disease. But another area of research that I think is really important for drug discovery is biomarkers. And these are biomarkers in the human population of the disease. both because this is a way to test the pathway that you plan to target and see how it correlates with genetics, how it correlates with radiological data of MRI in patients. We're very fortunate right now to have access to very large genetic databases. So what we think is happening, I think the blood is important, but what if I can start correlating this with genes? And I can find specific patients that this is more damaging than others, because maybe blood can leak in many people's brains, but make more damage on some than others. And this is what the data might be showing. So the biomarker research in patients, coupled with the animal models, can be very important for us to understand really what these pathways do, but also eventually stratify these patients. We think a lot about a cure, but cancer, I think, proved to us that there is not one single cure. Cancer is proving more and more that we have a lot of what we call personalized medicine, meaning that patients with specific mutations respond to specific drugs. Neurological diseases might not be that different. We might not have one cure for Alzheimer's disease. We might have different population of patients within that disease that might benefit more for one drug or a mix of other drugs. And I think a combination of animal modeling and biomarker research can help us stratify patients and understand mechanisms of disease.
Just to finish the discussion, and what I said about the various viruses and the bacteria that we're using, we are sometimes very unfortunate because they will not replicate in any animal because they're very much specialized on the human body. So in that research, we have really totally specialized on these new models in human diseases, and we're totally dependent on them because we cannot use a mouse that we can use as a preclinical test tube before we can go into patients. So we really have to be very innovative here to work with patient-derived material and develop new, especially also three-dimensional, culture dish experiments where we really can reconstruct, organs in a dish and can use them. And as we are working with many different viruses, we have to be, proficient in many different of these systems, and that's very fortunate to be here at Gladstone and have such a great environment that helps us to actually achieve that.
Thank you. Just to put a postscript on the discussion about the FDA, you may not understand that there's actually two steps where the FDA comes into play for us, and I believe that the Gladstone philosophy is really the right one here. The first step is where you want the FDA to allow you to test your new idea, your drug. It's never been given to a live human before, but you need approval to test it. And that's where the Gladstone approach is to present an overwhelming body of evidence that doesn't rely on a single animal model. It utilizes human cells. We want to tell the most compelling story, so the FDA will allow us to do it. The other step where the FDA, and this is where what's in the newspaper most of the time, is the actual approval of the drug, to let it out. It's being tested in humans. It looks to have some benefit, and will it be allowed to go on the market and be used broadly? I think the best approach there is not to worry too much about the FDA policies, but to make drugs that people really need and drugs that really work. And if you do those two things, I have no doubt they'll speed through the review process. Where you've got something marginal, it's marginal, but that's what we want to do. People really need it, and it really works. Yes, Blake.
I just wanted to add that to the question about the FDA, apparently Scott Gottlieb, who's the new director of the FDA, has been getting a lot of credit from the investment community about removing a lot of barriers that have been put in front of the drug companies for approving these drugs. And there's been a whole lot of excitement in the investment community that some of these drugs that have been held up in clinical trials are all of a sudden going to be released and be available to be used by the public. So that's supposed to be one of the best appointments that President Trump has made is Scott Gottlieb.
The rate of new drug approval had fallen to historical lows about 3 years ago, but it's really picked up. So there is more FDA approval recently than what we'd seen in recent years. Yes. Fred.
On the cardiology front, absent some intervention, do you think that humans are born with a genetic profile that in effect predetermines how many heartbeats they get and how rapidly that cardiac failure will occur? And is that sort of an area that would be a focus for determining when you might need to intervene to take care of that cardiac failure?
That's a great question. So I'll repeat it. It's the question is there a genetic contribution that we're all born with that plays into how our heart function is going to be throughout our lifespan? And the answer is yes. We know for sure it's very complicated across large populations, but we know for sure that there's certain diseases that are caused by abnormalities in your DNA that you're born with that affect the heart and all aspects of heart and blood vessel function, and they can cause you to have heart abnormalities when you're just a kid or a teenager or later in life. But that's really the minority. That's really a small sliver of the pie. We can learn a lot from those diseases, but the vast majority of cardiovascular disease in the broad population, there's a genetic component, and then there's a component of our environment, and they likely interact. And those genetic components aren't like one mutation that drives the whole thing that causes you to have heart failure. Those are rare. There are a collection of maybe small changes in your DNA that all have little contributions, but all add up together to make you have more risk for heart disease later in life than the others. And there's a big push to try to understand some of that so that we can start prescribing therapies that we know already work, like cholesterol lowering and blood pressure lowering therapies to patients much earlier, or put them on higher doses because we know later in life you're going to be predisposed to heart attacks or blood pressure. But we're learning a lot in this space. This is this kind of field of the people call personalized medicine. So this interaction between genetics and environment. So.
I think I heard in Fred's question a second dimension, though, is that as we get older, do our hearts wear out?
Yes, and so this gets into this notion of aging, right? And so we are not built to live forever, but I think that we've done great over the last 50 years to increase the healthy and productive and happy lifespan of people. So people are living longer, and they're also living better. And I can tell you, at least in the cardiovascular space, basic research that's been done in the laboratory that's predated the clinical application by decades has made major contributions to lifespan and, more importantly, quality of life. So I can tell you in research that was done in the 60s and the 70s that helped us understand how cholesterol is transported in the body, and which the Gladstone Institutes was founded upon many years ago, has made it into clinical practice now that we have very effective therapies to lower cholesterol, and we're discovering new ones. And these types of therapies, at least for the cardiovascular space, have actually increased life expectancy in the United States by 7 years over the past few decades. And we can do better, and we need to do better for heart failure, heart muscle diseases, but certainly we can move the needle forward. But yes, trying to understand the genetic determinants of how people age is a big area of interest, not just for heart disease, but also for neurodegenerative diseases and memory and functional status, risk of infections, cancer, and it's a burgeoning field. So it's a complicated question. We don't have all the answers yet, but we're trying to understand.
Yes, please.
This question is a little bit out of your wheelhouse, but you're kissing cousin, so I'm going to ask it anyway. You are an organization that is devoted to learning and pure science and good thinking. And we live in a country wherein some major players are much less devoted to that than you are. And I am really concerned about identifying organizations that effectively can combat some things with which I strongly disagree. So I'm asking, I know the Cato Institute, I know the Brookings Institute, now I'm reasonably informed. Who are the thinkers that move this cutting edge of science research forward as distinct from trying to stomp it down and put it in the box and call it all the kinds of silly names that we hear too much of? And I'm guessing that you are closer to having a sense of that than certainly I am. I'm hoping would be the more fervent verb. So if you could wrestle with that, I would be most appreciative.
Well, I have an opinion on that, but I'll defer to my colleagues first.
Does anyone else want to take a crack at that one? All right.
I can only tell you personally, I'm unfazed. I think what we're doing is extremely important, and I think we will not have anything stand in our way to make it happen. I think this is probably something that unites all, the whole science, community. We are really very passionate about what we're doing. We're teaching young people to become rigorous scientists and carry the baton forward. And I think we will not stop doing this on a personal and an institutional level.
I would have 3 specific recommendations for you, okay? You may want to write these down. An organization that's nonpartisan, but it represents broadly science in America very well, is the American Association for the Advancement of Science, AAAS. It's based in Washington. Many of us are fellows of that organization or members. It's grassroots among scientists. The president actually was a member of Congress previously, Rush Holt. But it speaks responsibly and in a nonpartisan way about what science means for our country. American Association for the Advancement of Science, AAAS. You'll find it on the website easily. The second organization I'd point you to is Research America. Research America, again, is a not-for-profit, nonpartisan organization that really promotes the point of view that solutions to human problems will come through science, through rigorous science. We support that organization, and they're very credible and believable. And the third source would be the national academies. In Abraham Lincoln's presidency, he wanted science to be very close to governmental decisions, and he founded the National Academy of Sciences. That's now branched into 3 academies of sciences, medicine, and engineering. They have a website. They will have authoritative, nonpartisan, high reliability views on these issues. Several of us here are members of those academies.
You said there were 3:
medicine, engineering, and sciences. That's what they're called.
Yeah, so I just want to say a personal story that has carried me through when times are not as, as not as, usually supportive. I mean, we're very lucky here to be in a very supportive organization, and Sandy mentioned all the different agencies we work with. But as Melanie said, it comes down to your personal motivation and determination to rise above a lot of what's happening around you with this support. And the story is, I was very lucky in 2008 to meet Rita Levi-Montalcini. Rita Levi-Montalcini is one of the first women who won the Nobel Prize for the discovery of NGF. She discovered nerve growth factor. And she was 99 when I met her, and she came to present data at the meeting, at the international NGF meeting. She was not invited, but she contacted the president of the meeting to tell him, why haven't I been invited? And I have a new date and I want to come to present. So he emailed the speakers and said, I think we found a keynote lecturer. So she came to the meeting at age 99. And one of the questions that we asked her, again, it was a time that the funding was really bad in the States. And you remember we had the stimulus at the time. It was the stimulus of the government because of how bad the NIH funding had become. So we asked her, what advice can she give to scientists when funding is so bad? And she said to us, I don't know what you're complaining about. When I discovered NGF, I was fired from my job at the university. I was prosecuted by the Nazis. I had to work from my kitchen to do these experiments. Go do your job, and you will find the support that you need.
All right.
I have followed Rita's advice to not complain and not whine and to go do my job. And when the going gets tough, talk to Sandy and see how we can make this work, because we have to make this work. We owe it to the patients who depend on us, and we cannot let administration X and administration Y change the course of science. We do what we do, and with the support that we have from the environment around us, the Friends of Gladstone and the Friends of Science, is how we're going to be able to go to the next step.
Yeah. I just have to tell quickly another famous Gladstone story, because it's one of the greatest stories in all of science. There was a young man who grew up in a foreign country, wanted to become a scientist, and he sent applications to great places all over the world, and every place rejected him except Gladstone. So he came here, and he started to work on a project, and it went terribly. Everything he did failed. In fact, he had a new drug that they were all excited in, and they wanted to test it in mice, and the mice all died. And this was after he'd worked for a year and a half, so it wasn't looking very good. But in this unexpected pathway that I'm describing, he said, why did these mice die? Well, they died because the drug gave them liver cancer. And he said to himself, if a drug can go into a cell and take a normal cell and turn it into a cancer cell, why couldn't we send signals to cells that would change them to do something that we want them to do? And many of you have guessed by now that this is the great Shinya Yamanaka, who later won the Nobel Prize for discovering just that. It took many years between the idea and the proof, but he proved that we can send signals to cells and turn them into what we want them to be. But that's that perseverance in the face of obstacles, never giving up, and taking an unexpected path. That's what we do here.
Sandy, what's the update on Shinya's work as it relates to cell development in humans?
Let me answer that quickly, Will, because I want to give this other gentleman a chance. Shinya's work moved into humans right off the bat. Once he showed that he could turn a mouse skin cell into an iPS cell, the equivalent of an embryonic stem cell, he quickly did it in humans. So Shinya's work took that into humans right early on, and that's what created the ability that we have to create these beautiful models of disease using real human cells that are genetically identical to the living person they came from. But if what you're asking is, where is that now reaching the clinic? It's starting to do that. We give a prize every year for scientists who are making the boldest moves around the world to get that work into the clinic, and we'll be making that award next month. Yes, sir.
I have a rare disease question for Melanie, and if I may, a common denominator question for SAP.
Please.
Melanie, one group of diseases, albeit very rare, that might be worth trying to look at to learn more about that surveillance mechanism, that universal surveillance mechanism, would be the prion diseases. Prion diseases are rapidly progressive processes in the brain which turned out to be caused by infectious particles that are simply misfolded proteins, proteins that our brains produce, but perhaps for genetic reasons or mutation or whatever, when they're misfolded, and it doesn't happen until a certain age, and then it progresses very rapidly. And a relative of mine recently succumbed to one of those prion diseases, CJD, Creutzfeldt-Jakob disease. It would be difficult to study, to look for a surveillance mechanism, and I was going to ask you, why doesn't the surveillance mechanism recognize this abnormality, or do you think it could, but perhaps because it's in the central nervous system, perhaps the surveillance system is different there from where it is everywhere else in the body? I don't know. But anyway, it'd be difficult to study, one, because it's rare. Only about 300 patients are diagnosed with such a disease, so it's like a 1 in a million chance of coming down with it. But it rapidly, causes such dropout of neurons that the brain winds up sort of sponge-like. Bovine spongiform encephalopathy, or mad cow disease, was one form of this. So I'll just stop there and see if you have any comments with regard to this surveillance system. And in other words, the most important question is, could we detect some defense mechanism that the surveillance system tries to turn on, but it's inadequate, that we might be able to enhance?
Exactly, yeah. Yeah, that's a very good question. So the question is to actually expand our efforts, not only, from going for viruses or bacteria, which are, pathogens that live on having basically a nucleic acid that is, making them replicate and persist in our body, but also extend it to basically these prion diseases, which are also infectious, but are actually transferred by proteins directly. And it basically initiates a cascade of events just by the interaction of these proteins. And I think that's an excellent suggestion. We have not included these in our, current efforts, but we definitely have fantastic expertise here on campus, of course, to include, this in our future efforts. I think the key here is to really focus on the misfolded protein, which we cannot really fully reproduce with the current strategy that we are currently, following. But I think I see no reason why we cannot include this. Now, the brain is definitely a special organ in terms of defense mechanisms and surveillance. But as I pointed out, we are what we are doing is we actually making the utmost effort to go into the best model system in the dish to reproduce these changes. So we would definitely go into probably an IPS-derived neuronal model to look after these mechanisms. But it could be done, but it's just a little bit more complicated technically.
Katerina should speak to this too.
Yeah, so I think that, the question you raise about surveillance and prion diseases, propagation of prions, I think, is a matter of huge debate in science. We don't really know how they propagate in the brain and what are the immune mechanisms that actually control them. What I find fascinating in these diseases is, again, how these prion molecules and these misfolded proteins, like you mentioned them, are also triggers for blood-brain barrier to open the brain. And this might facilitate these immune functions and the surveillance that you mentioned to accelerate these mechanisms of immune surveillance of the prions. Something that is now an emerging field in neurological diseases is how these misfolded proteins in the brain, not only prions, but also in the case of synuclein, that is one of the main misfolded proteins in Parkinson's disease, how these proteins become new antigens that our immune system responds to. There is a beautiful study in Parkinson's disease about autoimmune responses to these misfolded proteins. So suddenly, this converts brain diseases that we traditionally considered just neurodiseases, it transforms them to being immunological diseases, meaning that our immune system is now the culprit of responses to these misfolded proteins. And again, finding which patients are susceptible to building up their immune system to these misfolded proteins will be very important for treatments. So this, there is this, the fact that I think Melanie and I are answering the same question shows to you how there is this reassessment of neurological diseases, that they don't have any more a single cause. There is nothing that is only neuronal or only immune or only vascular. This meet in the middle, and when we really start thinking the diseases through this prism, we might have a good chance of actually treating them.
I think we have time for one more question before we go to our reception. Yes, sir. Oh, I'm sorry. Someone have a microphone? Yeah.
Oh, is it? Sorry,
Here we are.
I couldn't see the hand.
Hi, I'm just interested. You guys make a great discovery and all that. Is there any relationship? How do you get that into the marketplace or into the hands of the consumer? Is there any relationship with the Gladstone Institute and our local biotech firms and all that?
Quite a bit. We have some people actively engaged in that right now here. Yeah.
I think each one of us up here has an answer to your question. It's a really important question, and it's something that research organizations and universities have been trying to tackle for a long time, is that how do we take fundamental discoveries made in the laboratories and try to move them forward? And I'll second what Sandy had said, is what we try to do first and foremost is try to do the most rigorous science that's convincing to show that whatever biology that we're studying is actually working the way we think it works and is reproducible and is potent, and we really figure out how things are put together and how they work. And then the second step of that is how do we move it forward, this process of translation. And I think that's a really complicated issue, but I think the single word there is partnership. And it has to be done through multiple parties with a common goal of getting drugs into patients. And it takes a big team, but I think the key is that we take discoveries and we work with funders, both private and public, as well as for-profit companies that want to move things forward. And I'll leave it at that and let others answer.
All right, would you like to do that now?
Yes. As a former representative of the William K. Bose Jr. Foundation, who has studied many companies similar to yours, one of the greatest assets I've found is the way the Gladstone Institute operates within a business-like atmosphere to progress and move product forward into a sensible market environment as quickly as possible. And that was one of the reasons why Bill was so interested in this company, and Bill was the biotech, he founded Amgen. So I really say to you, to answer your question, I think that they do a superior job to any other company that I know of myself personally in the United States with regard to moving these products forward. Thank
you. Yeah. Other comments?
Yes,
Yeah.
I thought this was very, thank you for your comment.
Thank you, Chuck.
It was, I just wanted to say that, yes, we have a dedicated, team here at Gladstone that actually facilitates these interactions. And I think that's very, very helpful for us. But I also wanted to point out that what you were saying to us being here as a nonprofit organization and there being the pharma companies becoming interested in something that we're doing, I think in between there is a valley of death where many good inventions die. And I think this, one of our, dedicated goals here at Gladstone is to bridge this valley of death and actually find new and creative ways how we can go from a basic discovery to a marketable product. And they are diverse, but we're investing a lot of time and effort in making this a priority.
Katerina? Katerina's right in the middle of this.
Yeah, so I would like to say that I've been at Gladstone now for 10 years. And when I moved here, one of the reasons that I moved my lab and what attracted me at Gladstone was the commitment to translation and the commitment to commercializing research for the purpose of taking drugs into patients. But commitment is not enough. You really need to have mechanisms in place to actually take academics like myself and teach them the skills and the language that we need to be able to speak to big pharma, to speak to investors, to pitch our ideas, and to start our companies. And Gladstone has the infrastructure in place through the translational office to actually take research from our lab that is at the right stage, when we develop it at the right stage, to be able to do the key experiments and speak to the people that is a broad community. It's a community of venture capital, a community of philanthropists, a community of big pharma that will help us bridge the gap and bridge the valley of death that Melanie said. So these things don't stay in our lab, in our mice, but actually find the path to the clinic. So Gladstone is not only committed, but also has the infrastructure in place and support of this infrastructure to be able to reach through more of what we do and take things at what we call early stage and de-risk them to the extent that will make them competitive for investment from the outside community and later lead to their commercialization. I think is what Gladstone is doing really well.
Yeah, we're about to conclude this part of the program. I guess I'd wrap this part up by pointing to our mission here, Science Overcoming Disease. That has two parts. It won't happen unless you do the science, and we start with that. But the overcoming disease part, we take very seriously. We don't just throw stuff out there and hope it happens. We're very purposeful by trying to get it in a path. And we recognize that our discoveries will never benefit patients and humans unless they pass through biotech and pharma and medical device companies. So we need those folks, and we interact with them very fluidly. So before we close, there's a number of people who need to be thanked for this event tonight. Quite a number of Gladstone departments made this happen. Our communications department, our information technology department, our philanthropy department, our facilities department. I want the representatives of those groups who are here to raise your hands so we can give you a word of thanks. Okay. And then I want to thank all of you again for being such loyal friends and for helping support us so that all of this great stuff happens. So now let's have a drink.